Hello, it's your boy, the one and only Sir Patrick of the Department of Biology. So, before I start lecture 2, I'd like to say some announcement for the laboratory portion of this course. So, I have decided that for our laboratory portion, I'll upload a couple, and by couple, I mean a lot of high-definition photos for you to browse upon, so that you have a sense of what we did back then. So I'll upload a lot of the specimens that we're using here in the campus, and I'll upload all of them in the Eyyy course site. So what you will need to do is to browse upon them and to start memorizing all of them. And by memorize, I mean memorize. And toto memorize. Yes. From the family to the genus and species epithet level, no? Species level, yeah. So, specific epithet level. So, yeah, because in biology, we believe that for a student to integrate, he or she must first know, no? What is there to know? Because you can integrate something if you don't know the basic parts to integrate, no? So, that's the thing that we'll do for the laboratory portion. So, I need you to understand that for the laboratory portion, we have two classes of organism. Two types of organism. We have the microalgae and the macroalgae. So for the microalgae, I'll upload some photos of mine that I have identified back then, and I'll use a PowerPoint for that, so you can browse on that and try to identify as much as possible all of them. So I'll try to use that field work that we did back then on the microalgae. So this is from a freshwater source, and we found a lot of microalgae on that small pond. So that's what you're going to do for the microalgae portion. Now for the macroalgae portion, I'll hire our RAs. I'll ask them to do some picture taking activities for you to appreciate those precious specimens that we have here. So I'll let them take a lot of pictures. I'll make sure that some details are clear on those pictures, clear enough for you to use them in the exam portion. So during the exams, I'll show the specimen. So we have a lot of specimen here, and I'll show them to you. So it will be an oral examination for the laboratory portion. So I'll pick maybe 5 to 10 specimens. I'll show them to you, and you will ID. You'll identify what they are. And if you succeeded in identifying it, you pass. And if you don't, you get deductions from your score. So that's the thing that we will do. So start memorizing all of those specimens that I will upload in the VLE course site. So yeah, better check that out soon. I'll upload it soon. And no, without further ado, welcome to lecture 2, Morphology and Ecological Diversity of your algae. So before we start our lecture for today, we'll now first discuss the outline. Our last lecture, we talked about Phycology as a science, then we move on the general characteristics of algae. We discuss what types of cells are found on this group, and moved on to the plethora of structures of your basic algal cell. But now, for our lecture of today, we'll focus on the morphological and ecological diversity of various taxa under the group algae. In terms of morphological diversity, the Polypalatic Group algae does not disappoint. We have unicellular representatives like your Pacus, again with a prominent stigma, and the one that we encountered earlier, like your Euglena, the one with rhodopsin, and also a stigma. And a very elaborate and ornate microsterias, that is a very close representatives of your Desmidium, a unicellular green algae of the order Desmidiales. And you can see its nucleus right here, displaying a bilateral symmetry with two mirror image of its cell and a narrow isthmus containing the nucleus of the organism. This one's a green algae. We also have representatives that are in colonies. Now, what do we mean when we say colonies? Colonies are assemblage of cells. Bear in mind that colonies is not synonymous to your multicellular organism. When we say multicellular organism, the difference between a multicellular organism and a colony is that your multicellular organism, when you pick one cell out of that organ, for example, that one cell cannot live on its own. Why? While for your colony, when you remove one cell from the colony, it can live on its own. That's the main difference between them. So, for example, when you pick out a brain cell out of a brain, it cannot survive on its own. Only if you provide it the right laboratory condition can it survive. But on its own, in the wild, a brain cell cannot live. But for the case of your colonies, when you remove one member out of the colony, that member can live on its own. So there are three types of colonies here. The first type that we'll discuss is what we call the cenobic type of colony. When we say cenobium or cenobic colony, these colonies have a specific number of cells in that colony. For example, letter D here, your motile cenobic colonial algae, like your Platydorina, has a certain number of cells when they reach maturity. So often, your cenobic type of colony has what we call, as a property that we call uteli or uteli. This is the condition wherein when your cell or when your colony reach a certain maturity, they have a fixed number of cells. Okay, so these types of colony have a specific number of cells, kind of like your C. elegans. Remember from your Zoo 111? Your C. elegans has a certain number of somatic cell when they reach maturity. So it's a very good model organism for genetics because of that. So for the case of your algae representative, they could also be a good model organism for most cellular biology experiment, because most cenobic algae representative have a fixed number of cells when they reach maturity. Aside from your Platydorina, we also have your Volvox, wherein there is at most 20,000 cells in that Volvocine community that perform photosynthesis. Of course, we also have non-cenobic colony. When we say non-cenobic, of course, this doesn't have a fixed number of cells when they reach maturity. So examples are your Microcystis, that is non-cenobic but still forms algal colonies. So it is this one right here. So the number of cells that forms this colony is indefinite. So it's not fixed. Okay, another form of your cenobic example of your cenobic but non-motile colony is your Pediastrum right here. So right here, letter C. So this is the one that is completely weird and completely arty. So it's kind of like an art form when you look at it. You can frame these images and put it in your drywall or something. It's a good art artwork for me. I think so. Yeah, it's a very good artwork. It has a very star-like pattern. So that's your pediastrum. Also, this is a cenobic colonial algae. So certain species has a certain number of cells, and you can actually use that to identify these organisms. And there are your linear types of colonies. Next is your filamentous organism. So for your filamentous organism, these are tubular, elongated, or thread-like forms of your algae that are formed due to perpendicular cell division. Perpendicular in reference to the long axis of the cell. So the division is usually perpendicular to the long axis of the cell. So if this is the long axis of the cell, the division would be perpendicular to that. And at the end of the cellular division, the daughter cells are attached together in a single chain. So when this one grows again and divide, they are attached to what we call the transverse cell wall. Transverse cell wall. So since they are attached via the transverse cell wall, they form a elongated single chain thread-like structure. That's why they are filamentous in nature. Of course, we have what we call the branch types of filamentous organism, and we also have the unbranched. Very simple. And also we have what we call the pseudo branch. So we have at least three. We have a branch, unbranched, and pseudo branch. We also have what we call uniseriate, and we also have the multiseriate. So let's first define this to explain more on what we mean by this. So under the filamentous algae, you can have a uniseriate or a multiseriate. So when we mean what do we mean by uniseriate? That's just single layer, okay? Single layer. That's when we say seriate, it's just a single layer of cells. When you have a multicellular seriate, you have multiple layers, 2 or more layers of your cell, those single chain cell. So that's your multiseriate na type. When you say branch, you have a branching pattern, of course. When you have an unbranched, usually this organism only forms a single chain of your filaments. But when you say pseudo branch or false branching, the way that the organism is branched gives the sense, gives the appearance of branching. This is not a true branching, wherein the branch arrangement of filaments are due to the protoplasmic bifurcation or cell growth, or the fission along more than one axis. The branching here, like for example in your let's go back. Like for example in your Cytonema, the branching is not a true branching because it's not due to the real protoplasmic bifurcation or the real cell growth. It's due to, usually it is due to a death of the cell or the slipping at the end of one cell past one another following the cell division. So usually false branching looks like this, while like this one, okay, that's false branching in your Cytonema, that's false branching. let's just put it right here, false. When we say true branching or the real branching, it looks something like this. So your cell grows like that. So the daughter cell divides and joins together to the transverse cell, and then they branch like this. So right here, you can see there is a real protoplasmic bifurcation and cell growth fission along more than one or more axes. But here, there is no such thing as that. The branching in your false branching is a result of a sleeping or a death of the cell at the end, no? And then the cell right here just continues to grow and creates that appearance of a branching, although it's not true branching. It's not due to the actual cell, no, having a line of bifurcation. Okay, so examples here are your uniseriate unbranching filamentous alga, like in your mucosa, just form single cell chains. Okay, that's uniseriate. We also have your unbranching filamentous alga right here, letter B, oligonium. It's like in your mucosa, your oligonium is also unbranching and thus forms long thread-like filamentous cells. But for the case of your hygieoclonium right here, it forms branches, single uniseriate branches. So for your Synocytic or your Syponous types of algae, there is a tubular filaments. No, this usually looks like a tubular filament that lacks transverse cell wall. What do we mean by that? The Synocyte or the Syponous organism usually have large multinucleated cell, and since they lack or they have an absence of that cell wall or that transverse cell wall, these Synocytes or the Syponous organism usually just looks like a continuous line of aggregates of nucleus that are not divided by a cell wall. So they are multinucleated because each cell, each nucleus, is not separated by a cell wall. So they just clump together in that singular tubular filament. Okay, so this is usually considered as a single cell. So they are one cell. Okay, they are one cell, but has many nucleus. Although this type of organism, this type of form, morphological form, is not that common in the algae world, but there are few representative, like your Vaucheria. Okay, so this Vaucheria species, looks like when you see a rock right there. So it's a very good drawing of a rock, if I say so myself. So if you see a rock, usually your Vaucheria species would grow or creep along that rock, forming a continuous cell. So this one right here is just one continuous cell with many nucleus in it. Okay, so those are your Synocyte or your Syponous organism. Another example is this one right here. This one is a very good example of a Synocyte or a Syponous organism. This one right here is actually one of my favorite one. This is what we call the acetabularia. Acetabularia. This is again just one cell with many nucleus, of course. And acetabularia is a particularly close genus to my heart because they are very easy to identify when you're in the field. Because look at them. Who would mistake them for anything else, right? So acetabularia is a genus of green algae, and their name comes from the acetabularia in Latin world, acetabulum, no? Which means broad and narrow, shallow, broad but shallow cup, no? Usually back then it was used for dipping bread in that shallow cup. Yes. So your acetabularia genus right here is again a very good cell, a very good model organism for cell biology, because again, this is just one cell, so you can experiment on it. So it's although it's gigantic in size, it's still just one cell. So it's a very excellent model organism for studying cell biology. The common name that we call this is mermaid's wine glass, because it looks like a fancy wine glass of your mermaid man and your barnacle boy. So yeah, so that's your acetabularia, again under the Synocytic or Siphonous organism, no? Forms. Now moving on. Moving on. So lastly, we have your, can you still see this one? Okay, so lastly we have your Parenchymatus organism. So when we say Parenchymatus, these are assemblage of undifferentiated cell. Okay, should be this cell came from the original meristematic totipotent cell. So although they are macroscopic in size, no, their cells, they're just a lump of cells, a lump of undifferentiated cell. Okay, and their division is usually in 3D manner. No, their division, as you can see here, looks like a lump. No, they look like a lump of cell, undifferentiated cell, just like your parenchyma in your botany. So the division usually is in 3-dimensional way: up, down, bottom, left and right. Okay, bottom, top, ganon. So yeah, the 3D division of your meristematic cell. So there really looks like lumps of Parenchymatus or meristematic cell. So examples of these Parenchymatus forms are your monostroma, and not to mix them up with what we call pseudoparenchymatous organism, no, pseudoparenchymatous form, like this one right here. This is your Enteromorpha intestinalis. So your Enteromorpha intestinalis is not a Parenchymatus type of morphology, no, in your algae. Okay, it's not a lump of cell. These are Filamentous, actually, Filamentous type of your algae, but they look like a undifferentiated cell, right? If you can see right here, this intestinalis here, or this one right here, they look like the first one that we saw earlier. They look like this, no? But they're not the same in morphology. This one right here is Filamentous in form. Okay, this one is Filamentous in form. The other one is Parenchymatus in form, but they look kind of the same, right? So yeah, better watch out for your Enteromorpha intestinalis in the field. A synonymous form of your Enteromorpha intestinalis is also your Ulva lactuca. Sometimes they are mistaken for each other. So yeah, this one naman is flat, no? Flat in nature. But again, these are not Parenchymatus in nature, because they are already differentiated type of cell. Okay, and they do not remain parenchymatous in nature or meristematic in nature. So that's why we consider them pseudoparenchymatous. They look like a undifferentiated type of cell, but they are actually differentiated type of cell, no? This one pa nga is filamentous in nature. Okay, so those are your better watch out types of morphology in your algae. Okay, now that we're done with the general characteristics of algae, we'll now move on to part 3, the general modes of nutrition. Here we'll discuss the 4 types of the general modes of nutrition. We have the autotrophic, the heterotrophic, the oxotropic, and the mixotropic types of mode of nutrition for your various representative of your algae. Now, we'll start with autotrophic. When we say autotrophic organism, what comes to your mind when you hear the word autotrophic? Okay, so when you say autotrophic, this should come to your mind that these organisms are producers. From the word auto, self, and then the tropic means food or nourishment. So they are self-nourishing or self-eating, kind of, no? So they are self-nourishing. They are producers, no? So they use inorganic compounds. So they use inorganic compounds, the carbon dioxide in your air, as a source of carbon. Okay, for example. So they use that carbon dioxide or other inorganic compounds as a source of carbon, and they will synthesize that carbon dioxide to form your sugars as their food. Okay, to form carbohydrates or sugars. So we have at least 2 types of your autotrophic organism. We have the photoautotropic and we have your chemoautotropic. The main difference is with the name. So for your photoautotropic, so they are also self-eating or self-nourishing organism, the same as your chemoautotropic. They are self-nourishing. The difference is the first one uses light to fix carbon. So of course they have an inorganic source of carbon. So the first one uses light to fix that carbon into sugar. The second one uses inorganic energy source. Okay, like your hydrogen sulfide or your methane gas, for example. So they use this inorganic energy source to fix that carbon dioxide, okay, to form your sugar. So they use that. Okay, so lighter energy, inorganic compounds for the second one. So yeah, for your chemoautotropic organism, remember this energy source right here, your hydrogen sulfide or your methane, can be oxidized, no? You can get electrons from this energy source. So when you use those electrons, you funnel it to your sugar, you can produce ATP. Okay? So you use the electrons from this energy source, your methane and hydrogen sulfide, to funnel it to fix that carbon dioxide into that sugar. So that's what these chemoautotropic organisms are doing. Okay? Now, moving on to the heterotrophic types of organism. When we say heterotrophic organism, what do we mean by this? These are us. These are the consumers. These are the consumers, okay? So these are organotropic in nature in the sense that they use organic compound that was already synthesized by other organism, no? Produced by other organism for their own good, for their food. Okay? So they use the organic compounds synthesized by other producers, like the first one that we mentioned, as their food. Okay? So there are two types of organism right here. We have the photoheterotrophic organism, and we have your chemoheterotrophic types of organism. Again, when we say heterotrophic, remember this, even in your Bio 120 classes, when you say heterotrophic, they obtain the energy, no? They obtain the food from other producers. They can't produce it on their own. So they need to get it from somewhere else. Okay? So what they will do, these types of organism right here, for your photoheterotrophic, no? They use the light naman to digest their food. So again, they are heterotrophic because they get the food, organic compound, from somewhere or somewhere else. Okay? Organic, unlike your autotrophic, okay? Pag autotrophic, we mean carbon dioxide, inorganic carbon source. When we say heterotrophic, organic carbon source. Now, for your photoheterotrophic, again, for your photoheterotrophic, they use light to digest the food, the organic food that they have eaten. For the chemoheterotrophic, they also eat the food, no? But they use chemical energy to digest the food. The same pathway to produce ATP as your autotrophic organism that we mentioned earlier. The oxidation reaction that we have. Okay? So yeah, your organotrophs or your heterotrophic organism can also be what we call phagocytic, osmotropic, saprophytic, and parasitic in nature. What do we mean by these terms? When we say phagocytic or holozoic, these are your raptorial organism. When we say raptorial, just like the Jurassic Park raptors, they eat the whole organism. They eat whole organism. These are like hunter organisms. Hunter, Sigon at Sikolua. Yeah, Sigorapika and Liorio. Okay, so yeah, these are raptorial organism. They phagocytosize the whole organism. When we say osmotropic naman, these are the pinocytotic organism. They can eat, but only the smaller and more liquid in nature food. Okay? When you say saprotropic in nature, of course, this feeds on the dead organism. When we say parasitic heterotrophic organism, these naman benefit from feeding off their host. Okay? Just like for your parasites of your red dots, when you encountered a coffee plant and a mango plant that has, so imagine this is the leaf. I know it's not a good leaf drawing, but imagine this is a leaf. Yeah, something like that. I don't know how I passed biology courses, but imagine this is a leaf. Let's put some, yeah. So for your coffee and mango plant, when you observe red dots, red to orange dots in their leaves, these are usually, or in their fruits, these are usually caused by species of algae. So these are parasitic types of your algae. Okay, so they benefit from the host. Okay, and also your just like your the one that causes malaria, right? Remember from our reading, the Apicomplexan Plasmodium falciparum. Yeah, so heterotrophic organism can be like this too. Now moving on to the next type. These two naman, this one and this one, these are what we call the special bulalo, the special type of your general modes of nutrition for this algae group, no? Because for your autotrophic organism, oxotropic organism, they are also autotrophic in nature. Okay, your oxotropic organism are also autotrophic in nature. They are autotrophic. They absorb, however, they absorb organic material as vitamins. They still require organic compounds, not as a carbon source or not as an energy source, because they are autotrophic. Okay, not as an energy source, but as a vitamin source. So they need to ingest other organic material as vitamins, as supplement for their growth. So they need to ingest this organic material as a source of your vitamins, as a source of biotin, thiamine, and your cobalamins. So as a source of vitamins. So as a source of vitamins. So they need to ingest food as a support for their nourishment, not as an energy source. Okay? Because they are autotrophic, they can produce their own food, but they need to ingest this just to get those nasty vitamins. So they are oxotropic organisms. Now, we also have your other type, your autotrophic mixotropic algae. So these are also autotrophic algae, but they are facultative in nature. What do we mean by facultative? So again, from your Bio 120 classes, when you hear the word facultative, they can switch from what mode of nutrition they want. They can be heterotrophic or they can be an autotrophic. For example, are your euglena. That's why we're always mentioning euglena earlier on in the lecture, because this bish right here, yeah, this one right here is a facultative heterotroph. They may choose depending on the environment. They can be autotrophic or they can be facultative heterotrophs. Okay, so those are mixotropic organisms. Another example would be the one right here. So this is your Gymnodinium. This is a dinozoan member. So Gymnodinium is of the genus of your dinoflagellates. It's a marine freshwater plankton. So it's a naked dinoflagellate, but it's a dinozoan that feeds on. So Gymnodinium feeds on. So it's a dinozoan that feeds on another algae. So it feeds on a golden algae, your Rhodomonas. That's why it has this brown golden color to it, because it feeds on Rhodomonas salina. So Gymnodinium, a dinozoan, a dinoflagellate, feeds on another algae, a golden algal, your Rhodomonas salina. So when it feeds on this algae, of course, it will digest the other cellular structure of the algae, but then it will leave the chloroplast. No, it will not digest the chloroplast right away. Okay, so when your this dinoflagellate, this Gymnodinium, eats the Rhodomonas salina, no, it will digest the Rhodomonas salina, but it will leave the plastid, the golden tinged plastid on its own. So that's why it has a golden brown color to it. But this time, it will kinda use those plastid and become an autotrophic organism. Okay, so it's kind of weird. So as Patrick Killing told you, these dinoflagellates are really weird types of organism. They can acquire, they can lose, and then acquire again new plastid. That's why they are pretty weird in terms of their endosymbiotic history. So why they are the weirdest group. They are prone to controversies actually. So this Gymnodinium is what we call a klepto. So magnanakaw, no? Klepto chloroplast. So they stole chloroplast from other organism to use it on their own for their own good. Okay, so again, Gymnodinium eats Rhodomonas salina. It will digest other parts of your Rhodomonas salina, but it will hold on to the plastid and will become autotrophic for some time. But when the plastid dies, it will eat again, eat another Rhodomonas, and then get their plastid again for their own good. So they are pretty interesting and pretty weird types of organism in terms of their mode of nutrition, if you look at them in that way. Now that we're done with the general modes of nutrition, we'll move on to the general modes of reproduction of your algae representative. So let's first compare and contrast the benefits of using an asexual mode of reproduction versus a sexual. Again, from your GE courses, from your Science 11 courses, your asexual mode of reproduction is good in the sense that it has low energy use, no? Low energy requirement. You just need to divide, that's all. You just need to grow and divide. Yeah? But the downside of your asexual mode of reproduction is that you have low variation. And again, we need variation for many things. We need variation for survival, because when you have varieties in your population ecosystem, your nature can choose what it wants, no? So it has more chances of winning the evolutionary lottery, no? It has more chances of surviving. So that's why your asexual mode of reproduction has also low rate of survival. But for the case of sexual reproduction, imagine the cost of energy. Okay, it's very expensive. You are putting all of your energy in one basket, literally putting all of your eggs in one basket. That's how it looks like, because you need to produce energy. You need to put in so much effort in that offspring, in that mode of reproduction. But the thing is, pwede pang masayang, no? Hindi siya laging gagana. You need to find the mate, you need to do the courtship ritual, you need to look gorgeous, you need to have high degree of comedic preferences, you need to read books or something else, kind of like that. But yeah, it's worth it in the end if you get to reproduce, because you have high level of variation, because again, due to meiosis, due to the recombinant DNA, and due to the alignment of your of your chromosome in the metaphase plate, no? They are completely random. That's why it has more variation, and of course, when you have high variation, it has high rate of survival. So we want to survive, that's why we need sex. Kind of like that. Okay, so yeah, let's look at how the algae groups are doing it here. Okay, so we'll first talk about the asexual modes of reproduction. We'll first go with your cellular bisection. And in this portion right here, we'll talk about the Pacus genus. So Pacus is a genus of unicellular excavates of your phylum Euglenozoa. So it looks like a flat lip-shaped organism. Okay, so it's a eukaryotic green, mostly green in color, and they divide by splitting their body in half, just like this. Okay, they split their body in half, and this will become 2 new cells, just like that. So your organism right here in Pacus cellularly bisect itself, no, through cellular binary fission via longitudinal division. Okay, so your cellular bisection can be of transverse and longitudinal. So how do you determine if it's transverse or longitudinal? Just look at the long axis of the cell. If it's perpendicular, that's transverse. If it's right with the long axis, that's longitudinal. Okay, the reference is the long axis. So this will be the format of these organisms. So another example here would be, again, your Euglena. Euglena also uses cellular bisection right in their stigma, no, right in the new flagella when they're bisect. So usually your cellular bisection are for your unicellular or organism. Okay, next would be spore formation. Let's first discuss your zoospore and aplanospore. When you say aplanospore, these are afflagellated, so this doesn't have flagella. Okay, what do we mean by flagella? These are the what we discussed earlier. Okay, the whiplash, the hairy structure, no, whiplash or non-hairy structure that we described earlier, and these are for motile purposes of your spore. So yeah, your aplanospore are afflagellated, but they can develop into a flagellated organism. They can develop into a flagellated organism, but they start out as an afflagellated organism. They don't have flagella at birth, but they can develop into a flagellated organism, for flagellated type. But your zoospore, on the other hand, has your flagella, meaning they are motile. So your aplanospore, since it starts out as an afflagellated organism, it can develop into a zoospore later on if it wants to. Okay, can develop into a motile type of organism. Okay, so yeah, so again, aplanospore, zoospore. So this is how it looks like when your spores are released. They grow inside that vegetative cell, but when the zoospore are released, they venture out in the wild and then settle on somewhere else. Your spore are technically asexual modes, asexual agents of reproduction. So your spores are produced or can develop from a normal vegetative cell or from a specialized structure called your sporangium. So they can either develop from a sporangium or a vegetative cell. Okay, so yeah, but basically these spores, now these 4 types of spores that we're talking about, these are asexual agents of reproduction. So your zoospores are motile, your aplanospores are afflagellated at first but can develop into a zoospore eventually. Your autospore, on the other hand, are afflagellated. No, they are flagellated. They are non-motile forever. No, there is no further development for your autospore. So they are non-motile whatsoever. Your monospore, on the other hand, looks like a single, just like a single monospore, or a simple non-motile asexual spore, just like your autospore. But your monospore are for most representatives of your rhodophytes. So the simple non-motile asexual spore of your rhodophyte genera or rhodophyte groups, we call them monospore. So these are for your red algae, okay? But they are also non-motile and asexual spore. So yeah, because most red algae representative commits the act of what we call alternation of generation, and your monospores are produced in that manner. So yeah, let's have a recap. So your planospore is flagellated. It is non-motile, but it can develop into a zoospore eventually. Your zoospore are motile, okay? Your autospore are just like your aplanospore, no? It's non-motile, but they lack the capacity to develop into zoospore. Your autospore are for your green algae. Okay, autospores are for green algae, while your monospores naman is for your red algae. But they are exactly the same in the sense that they are non-motile, they are flagellated, and they lack the capacity to develop into a zoospore, into a motile zoospore. Okay, now moving on, we'll now go to autocolony formation, wherein one cell is independent of each other and can produce a new colony. So how does this happen? How does this look like anyway? How does colony formation work? Usually, autocolony formation is seen on your synobic organism, synobic colonial organism. So wherein each cell in your synobic organism, like your Volvox and Pediastrum, remember your synobic organism like this has a certain, for example, your Volvox has only a cell limit, yeah? It has a cell limit of 22,000. Your Pediastrum, let's just say 20 or something, okay? Approximately 20 or something cell, depending on the species. So they have a certain number of cell when they reach maturity. So in a synobic organism, when the cells, of course, will continually undergo consistent number of successive division. No? When they reach a certain number of successive division, they will give rise eventually to a miniature version of the original synobium. Okay? So each cell in the colony actually has the capacity to produce another colony, a new colony, which is a miniature version of the parent. So for example, in this Volvox colony right here, so we call this one right here, which arises from a single cell again that only divides to a certain number. We call this one the micro colony. Okay, so this will eventually leave out of this lump here and will produce a new colony of itself. Okay, so for Pediastrum naman, after the cells and the original colonies reach a certain number of division, it will leave out and will produce a single miniature colony that resembles its parent. Okay, so yeah, autocolony formation are for your synobic colonial organism that has a certain number of cells, and they look astonishingly artsy. They look really good, no? Especially your Volvox. Look at this colony right here in the black background. Diba? Napakaganda. Okay. Next example that we'll talk about, and again, still under your asexual mode of reproduction, is your fragmentation. So for your fragmentation, this is simply a random process of fragmentation, of breaking apart of your non-cenobic colonies. So for non-cenobic colonies, or for filamentous algae, due to random process, you throw them apart, no? You break them apart and throw them away or something. They like that because they get to grow, no? They get to grow into separate organism. But this is a completely random process. Hindi nila ginusto na paghiwa-hiwalayin mo sila. So yeah, when you separate them, this one will form a new organism. This one fragment right here will form one organism. This one fragment right here will form another organism. This is for the case of your filamentous organism, like your here Spirogyra, because it has a spiral chloroplast. It looks very amazing too in the microscope when you see it. Yeah, so that's fragmentation process for you. Okay, and the last one that we'll talk about is your resting stage. So the first example of a resting stage that we'll talk about here is what we call your actinids. So your resting stage asexual modes of reproduction are essentially, when you say resting, this is a dormant stage in which the resting form of that organism can resist extreme environmental condition. Remember your lecture from your Bio 120. It looks like and it feels like an endospore of your bacteria. That's how your resting stage of your algae groups looks like, because they are dormant stage which can resist extreme condition. So resting stage type of asexual modes of reproduction are actually good for adaptation, for survival, because they can time the production of this resting stage at extreme condition, and they can survive that condition and let it pass through. Okay, so usually your resting stage forms are take hold in nature to enable them to resist the current environmental condition and to survive that environmental condition through that dormant stage. So there are different types of resting stage, but the first one that we'll talk about here is what we call the actinids. So your actinids are vegetative cells that develops a very thickened cell wall that is usually larger than the typical vegetative cell. So where's the actinid? Right here. So it's right here, here, and here. So as you can see here, it's very large, no, compared to the normal vegetative cell that you can see right here. And I didn't point out this one as a vegetative cell because that's not a vegetative cell. It's another form, no, that we'll talk about later on in the discussion, way forward in the discussion. But yeah, your actinids are right here. They are very large cell, no, it's abnormally large cell than your normal vegetative cell, because they contain large amounts of food reservoir, and they are enclosed by a very thickened cell wall. So your actinids of your Anabaena species, your Anabaena genus, the actinids of your Anabaena genus serves as a mechanism of survival. Okay? So other forms of your resting stages, so we have talked about the actinids. So this is your actinids right here of your Anabaena genus. So again, this is the one that I was talking about. This is not a regular cell, nor it is an akinete. It's of a completely different type of structure of your Anabaena genus. So these are your akinetes again, a very large and weird vegetative cell, a resting stage of your cell. So we have discussed that. We also need to discuss what we call frustules, no? Frustules. Statospores. So what are statospores naman? Ang dami naman ng mga spores na yan, kung ano-ano, sir. So your statospores naman are for your other organisms na magpo-form din ng resistant stage, no? Resistant resting stage. And your statospores are for your organisms that has frustules, usually for your dinoflagellates or your diatoms, no? So for diatoms, the centric diatoms, for example, this is what we call the frustules. So when this is the top view, no? Top view of the diatoms. But when you look at it on a side view, it looks something like this. So your diatoms looks like a petri dish when you look at it sideways, no? Side view. The side view of a diatom looks like a petri dish like this. So this is what we call the frustules. The structure, no? The case-like structure of your diatom. So your stratospores naman are located within that frustules, no? It looks like a spores inside that frustules. So yeah, it's inside. The statospores are formed inside that frustules naman. Okay, so that's for your diatoms. So we have the akinetes right here. We have the frustules inside that. We have the statospores inside the frustules, and we also have your hypnospores for the case naman of your Eulotrix. Okay, hypnospore for the case of Eulotrix. So usually for a filamentous algae like your Eulotrix, they divide and divide. So imagine this is your filamentous algae like your Eulotrix. So a hypnospore would look like something like this. So it's still a normal, sorry, medyo pangit yan. Still a normal uniseriate cell, still uniseriate, but on certain areas, certain region of your uniseriate Eulotrix, you will see a portion where it's kind of different in morphology, wherein it's more enlarged. It has many somewhat dense region. So this is your hypnospore. So eventually your hypnospores for Eulotrix can free up, no? When the condition is right, these hypnospores right here can free up and it will release those spores, and these spores will grow to a new Eulotrix, a new filamentous Eulotrix in the wild. Okay, a new Eulotrix in the wild. But yeah, it's a dense thickened structure again to resist that dormant resting stage, no? To survive that resting stage. Okay, so hypnospores is just like akinetes, but for your Eulotrix genus. Okay. Now, moving on to our sexual reproduction. So, for your sexual mode of reproduction, there are usually two ways. We have your plasmogamy and your karyogamy. What do we mean by that? Plasmogamy is the union of the plasma membrane, and the karyogamy is the union of your nucleus. That's just that. Fusion. Okay? Fusion ni Goku and Gohan and the other one, Trunks. I don't know. Okay, so there are three types according to the gametes that are formed. So we have your isogamous, and we have your anisogamous and oogamous. So we have 1, 2, and 3. So 3 types according to the gametes that are formed. So your isogamous gametes are both motile. So these are monomorphic. Okay, monomorphic gametes. When you say dimorphic gametes naman, like your heterogamous, there are difference between the gametes. We have usually a small and a large gametes. So that's heterogamous. There are 2 types of heterogamous gametes. We have your anisogamous and oogamous, in which both the small and the large are motile for your anisogamous. For your oogamous, only the smaller one is motile. The other one, the larger one is non-motile. That's for your oogamous. Okay? So, yep. Next, we need to discuss the difference between monoecious and dioecious. Well, in the realm of your algae, when you say monoecious, there are 2 types of gametes that may be produced by the same the 2 types of gametes may be produced by the same individual. Okay, so these are usually common in anisogamous and oogamous reproduction. So for your monoecious, the 2 types of gametes may be produced by the same individual. The two types of gametes produced by the same individual. Now for your dioecious, the 2 types of gametes naman, okay, 2 types of gametes. The 2 types of gametes are formed from 2 different individuals. Okay, so that's dioecious. So in 2 types of gametes, form from one indiv and another indiv. The 2 types of individual. Okay, again, your monoecious, the 2 types of gametes that merge together, may be produced by the same individual. Now for the dioecious, the 2 types of gametes are formed from 2 different individuals. The 2 gametes must fuse and form from 2 different individuals. Okay. Now, moving on to the homothallic and heterothallic, the difference is when you say homothallic, from the word homo, it is self-fertile. When you say heterothallic, that's again self, but this time it's self-sterile. Okay, self-sterile, self-fertile. What do we mean by self-sterile? So when the gametes from the same individuals, they are for your homothallic, your self-fertile, the gametes from the same individual are able to fuse and produce a viable offspring. Okay, so that's homothallic. The gametes from that one individual, can fuse together from that same individual. Again, the gametes that it produces can fuse together to produce a viable offspring. Now, for the heterothallic, the self-sterile, when the gametes from the same individual, so if this one produces 2 gametes, usually these 2 doesn't fuse together. It doesn't produce a viable offspring. Okay, so the gamete that is produced from that individual are incompatible. So the individuals, No, the individual must find, no, another individual. The individual. So that person or that organism must find another organism, no, of a different genetic makeup. So a different organism of different genetic makeup are needed and are required for a successful mating to occur. Okay, that's for your heterothallic. Now, for the last part of your sexual mode of reproduction, in terms of types of life history, there are three. Okay, so we have the zygotic, the gametic, and the sporic. Now, I want you to examine these three carefully and examine it on the lens and on the basis of these factors right here. Because usually, these types of life history under the sexual modes of reproduction are classified based on these three criterion right here. So the first one is the time or where the meiosis takes place or the nature of meiosis. The second one is the type of cell produced after meiosis. The third one is the presence or absence of more than one free living stage. Or for my case, what I call this is the dominant free living stage. Okay, so let's examine these three carefully on that context, on that lens. So these are the three. Okay, so let's first go to your haploid life cycle. Okay, so this is what we call the zygotic meiosis, also known as aka your haploid life cycle or your haplontic life cycle. So why it's called haploid life cycle? Because the majority, okay, of its vegetative stage, the most of their life, it is haploid N. Remember your science 11, right? Now for your diploid organism naman, or your gametic meiosis naman, or aka your diplontic or your diploid life cycle, the majority of its life, it is a 2N organism. It is a diploid organism. Now, for the other case, for the sporic meiosis naman, this is aka your haplodiplontic life cycle, because your organism has an alternation of generation between the N and the 2N individual. Let's put it right here. Alternation of generation. Okay, so the majority of its vegetative organism cycles or alternates between the N and the 2N individual. So that answers the one question that we're trying to answer, question number 3, the presence and the third criterion, the presence and absence of more than one free living stage or the dominant free living stage of that organism. For your zygotic meiosis, aka your haplontic life cycle, the haploid stage is the dominant one. For your diplontic life cycle or your gametic meiosis, the diploid stage is the dominant one. For the haplodiplontic life cycle, they alternate, no, from the N and the 2N individual. Okay? Now, let's now answer the question of the first criteria, the time where the meiosis take place or the nature of meiosis, because this is a more easier one than the second criteria, the nature of meiosis. So again, for your haplontic life cycle, what is the nature of meiosis? So where did meiosis takes place? So for your zygotic meiosis, for your haplontic, that's why it's called zygotic meiosis, because meiosis happens after the generation of zygote. Okay, that makes sense. That's why it's called zygotic meiosis, because it's in haploid life cycle, in haplontic life cycle, the meiosis happens after the generation of your zygospore, the zygote. Okay, meiosis happens right after the formation of your zygote, zygotic meiosis. For your diplontic or your diploid life cycle, meiosis happens right after, right after for the gametic. That's why it's called gametic, because meiosis occur in the reproductive structure of the thallus. Okay? In the reproductive structure of the thallus. So, meiosis occur in these gametes right here, in the reproductive structure right here of the thallus. Okay? We call this thallus, or these types of organism, the conceptacle. This is the conceptacle. This contains both the male and the female structure, the conceptacle. It has the spermatogonium and the oogonium. Okay? So that's why it's called gametic meiosis, because you have the oogonium here and the antheridia right here, the spermatogonium and the oogonium right here that was already formed. No? It's kind of like the gametes that's already formed. Okay, in that part of your in the conceptacle. Okay, now for the sporic meiosis in the haplodiplontic life cycle naman, that's why it's called sporic meiosis, because the meiosis takes place, no? The meiosis happens at the sporophyte generation. That's why it's sporic meiosis, because meiosis happens right here, right after the formation of your sporophyte, no? In the sporophyte generation itself, actually. So we have answered time where meiosis takes place, or the nature of the meiosis. For the haploid life cycle, it's zygotic, because meiosis happens right after the zygospore is formed. The zygote is formed, no? The diploid life cycle naman, it's gametic meiosis, because meiosis happened when on the conceptacle, where you can find both the oogonium and the antheridia, the spermatogonium and the oogonium. Okay? On the haplodiplontic, that's why it's called sporic meiosis, because meiosis happens on the sporophyte stage of the organism. Okay, last one. Let's erase this and let's answer number 2. Now, when you compare them naman on the type of cell produced after meiosis, when you look at them closely, you can see again the difference, no? So for your haploid or haplontic life cycle, no? What types of cells are produced after meiosis? Look at it. So here is the meiosis right here. What type of cell? What types of gametes are produced? So your types of gametes here are what we call, sure na ba kayo sa answer nyo? Anong answer nyo dito? Isogametes. Why is it isogametes? Because look at this too, right here. They look similar. They look similar and they are both flagellated. Those are isogametes. We have the negative and the positive one right here, just to demarcate them from each other. No, kind of like a male and a female. You can't tell. There's no such thing as male and female in the isogametes. Okay. So now for the diploid life cycle naman, look at it. What type of gametes, or what type of cells are produced after meiosis? Gametic or the gametic. What type of gametes? These are oogametes, right? So these are oogametes. Why is it an oogamete? The larger one is immotile, non-motile. The smaller one is motile. Again, these are heterogametes too, right? But it's specifically an oogamete because the larger one is non-motile, the smaller one is motile. Lastly, for the case of your haplodiplontic life cycle, in the case of Ulva lactuca, the gametes that are formed right after meiosis, although right after meiosis, you will still form the zoospore, and the zoospore will develop to your gametophytes, and the gametophytes will then produce the gametes. The gametes here now are what we call an isogametes too, because if you look at them right here and right here, they are both motile and they are monomorphic, meaning they look the same. No? They are same in size and they are both motile. So these are definitely an isogametes. Okay, so the species that we studied here, no, an example for your haplodiplontic, we have Ulva lactuca. An example for your diplohaplontic, a diplontic organism, we have your Fucus, a brown algae. And for your haplontic life cycle naman, we have your Chlamydomonas. Okay, again, we have isogametes, we have oogametes right here, and again have your isogametes here. Okay, so yeah, I hope you enjoyed the lecture for today. If you have further questions for Sir Patrick, don't forget to message me. You can message me through Messenger, through Gmail, at pfpenales@up.edu.ph. And you can also do a comment section and do your questions on the comment section right here down below. And please subscribe, like, and share. For more content, just tune in our VLE course site and this YouTube channel right here. And yep, that's all for today. This is Sir Patrick. Peace.